Recording.
Okay, today I'm going to cover how you should have notes and how you should study from them, both from a lecture presentation and from reading the textbook. And I'm going to start by going through what your notes should look like from lecture. So if you have an opportunity to print out the slideshow presentation from your professor, that can be really helpful in getting a good start on taking notes. Now, as I was going back through this lecture, I realized that there was quite a bit of knowledge that I expected students to have already. So in your higher level courses, this is going to be standard. So as you're going through the lecture, if there's something that you are not understanding, kind of double check that you are understanding some of the material that is assumed by the instructor. So in this case, I organized the things that make a living thing using this acronym CHARM. That's been introduced in my class, but if you haven't used it in your class, then that's complexity, homeostasis, adaptation, reproduction, and metabolism. You need to have a basic understanding of the 4 types of biological macromolecules. What are the differences between prokaryotic and eukaryotic cells? What natural selection is, what the central dogma is about using the information in the DNA in the cell, how cells do division, a little bit about population growth from an ecological perspective, how rapidly do populations grow, interspecific interactions, so things like predation, competition, and in this case, parasitism is a very important concept. And then a basic understanding of the diversity of life, kind of getting back to prokaryotic versus eukaryotic and animals, plants, fungi. And then basic knowledge of how cells form into tissues, tissues form into organs and organ systems, and the basic body organs in humans. All right, so if you feel like you're missing something, again, make sure there isn't any background knowledge that you don't know, and look that up somewhere. All right, so as we go through slides, we're going to look at what are the pieces of information that are conveyed in the lecture on each of these slides. And an important place to start with the outline is that this lecture is going to answer 4 different questions. What are viruses? What are the types of viruses? How do viruses replicate? And what happens in viral infections? It's useful to mark down on your notes which slides go with which of those questions, so that as you're formulating your answers to those, then you can make sure you're covering all the important context. So if there's a few simple facts that are presented on each of the slides, you want to make sure and note down those. Make sure you're understanding what's being shown in the picture, that this is the virus and this is a bacterial cell. There should be, or at least a good lecture, a good organization of a lecture, there should be 1 or 2 main points that are take-home messages. So on this slide, I talked about viruses are non-living. What does that mean? They're an intracellular parasite. They're not a cell, they're not an organism. And then I talked about naming conventions. The naming conventions part of it is just to reiterate this idea that they're non-living. If they were living things, they would have a normal Latin binomial name, and that this content that wasn't in your textbook is intended to help reinforce this main idea that's part of this slide. When we start looking at the next section of the lecture, the vocabulary that's important on the slide, make sure that things that are mentioned by your instructor are things that you've circled or highlighted or written down. If you didn't get their meaning, then you can go to the textbook. Make sure and look those up in the glossary. Now, your instructor, if they're a good instructor, in my opinion, may introduce some extra information into the lecture that is intended to help you better understand the content compared to what you might get from just reading the book. And I put this slide into this lecture because when I was thinking about what the viral genome consists of, it was a little confusing to me. I thought about what genes were being mentioned and wondered if there were more different ones. It turns out no, there's just not a lot to the viral genome. And so this is not in the lecture. This slide isn't in the lecture so that you memorize, the names of these genes or anything like that, but it's something that hopefully a good instructor will do in order to help you understand, to go a little beyond what's in the textbook, to help you understand what we mean when we say the viral genome. What are the proteins that it makes, and then what do they go on to do? All right, these next two slides have to do with the Baltimore classification, and there's a lot on here. In order to understand this, you need to understand how DNA is used to make messenger RNAs, and then the ribosome is going to turn that messenger RNA's message into a protein. And so if you're hazy on that process, none of this is going to make sense, and why it matters if the viral genome is DNA versus RNA, if it's single-stranded or double-stranded. And knowing what are the viral proteins compared to proteins that it's going to use from the host cell, it's important to know that there's normally a DNA polymerase that we find in all celled organisms. There's also an RNA polymerase. What do they do? What's sort of the normal function of those? And then we'll see that there are these weird things that viruses have to do, and that they need their own unique enzymes in order to make those things happen. All right, so a few key concepts hopefully you got from listening is what reverse transcriptase does, that it's going to take an RNA message and build a DNA message out of it. And that only happens in viruses. So that's a unique and interesting thing. That DNA then may insert into the host cell DNA. So that's one important take-home message from this slide. Now I've highlighted this slide, I've highlighted the next slide. It is an example of one of these Baltimore classifications. And if you are not feeling very clear on those, there's a resource added here that shows you all of the little diagrams like this for all of the rest of the types. So if you would find that helpful, it's there. The main point of this slide is to make clear that the viral genome is used to do two things. One is to make copies of the viral genome to go into new virions. And the other thing is the viral genome is used to make the proteins that are needed. And depending on the type of the genome, different processes or different enzymes may be needed in order to do those two different things. All right, the viral replication cycle, there's an outline here with the different steps. You should know those different steps. This is an example in this figure, just showing what does attachment mean? How does the virus attach to the cell? There's some kind of protein. And then different types of viruses based on their structure, based on their type of host, based on whether they have an envelope or don't have an envelope. That's going to influence how they do their replication cycle. So we have to know some things about the virus we're looking at in order to be able to say how it's going to go about doing its replication cycle. All right, so we have examples. Now these figures are straight from your textbook. There's the steps that are going to happen for an animal cell, disease such as influenza. There's steps that are going to happen for a bacteriophage infecting bacteria. And there's two parts to this process. There can be this lytic cycle or this lysogenic cycle. A good way to study or make sure that you are understanding these two types of replication and how they're different is to actually sketch, to practice sketching in some way these two different cycles and to show what is happening. Perhaps put the virus genome using a colored pencil or something to make it clear what's happening. All right, the last part of the lecture, it's a little lighter in content. So if you're thinking, oh my gosh, what am I supposed to learn from this section? Many of these questions may be sort of how your instructor might ask you questions, could be something like true or false questions. So this point is basically being made. All different organisms have viruses. So you could get a true false question, you know, only eukaryotes have viruses. That would be false. Plants don't have viruses. That would be false. And then again, this slide, it's not intended for you to memorize all the differences in the H's and N's that different organisms have. It's intended to help you understand how, especially how humans are going to get viruses, that they're going to be close enough to us from an organism that the virus will be able to switch to having a different host. And this gets back to the idea of host specificity and why it matters, how we can have different organisms that are infected by different viruses. All right. Same thing with this page, a little less content here. Basic idea, different viruses are going to have different symptoms depending on the body part that is infected. That will influence how it's transmitted. How diseases are transmitted is extremely important in human society, how we are going to be able to prevent different diseases. The easiest way to treat them is to prevent them in the first place. This slide didn't really say too much about this slide. Basically, there's many different viral structures. By looking at the structure, we can't really tell what symptoms it's going to have. So if you didn't feel like there was too much there, that was correct. And finally, what are the impacts of different viral diseases in history? And what has changed since humans have discovered and been using antibiotics, why it matters, because antibiotics don't treat viruses, and how are we going to protect ourselves from viral disease. So there's a little bit more covered in the lecture here than I managed to get down onto my notes, but there is more of this covered in the textbook, and also it's things that we may be more familiar with from talking about COVID. So hopefully it made sense to you. Maybe you knew some of that information already, and it wasn't something that you felt like you were needing to memorize as you went. Okay, so a couple things about taking notes on the lecture. Notes are not the same as writing down every word that the professor says. As you're listening to the lecture, you should be focusing on keywords and concepts, that's for sure. But part of the purpose of the lecture is to explain things so that you understand them. If you understand them, then you don't have to memorize them because you understand them. You could sort of build them up again in your mind from things that you know, right? So if you really understand the idea that viruses have very specific hosts, but with small mutations, they might be able to infect a different host. This diagram is basically showing that concept. So if you understand that concept, if your instructor gave you a diagram that was new or different, you should be able to apply that understanding. All right, so taking notes from the lecture may not necessarily feel like you really have kind of a good outline to study from, and that is where the textbook can really help. So whether you read the textbook before you listen to the lecture or do a little bit before and a little bit after, both, that is going to help you really get the material organized, because as you're reading the book, you can write down all the pieces of information that are in there and then see what the organization is like. So the very first little section of the textbook, 21.1, I ended up with quite a lot of notes on. So you're able to write down when you're going through the book a lot of information. Make sure the vocabulary is written down, that you have some key facts, like there's no fossils of viruses. That is going to help explain why we don't have a taxonomy of them. Some of the things that are important in telling different kinds of viruses apart. Notice that I've got, a list of different things. At this point in reading the textbook, I didn't really understand what enveloped meant. So I made sure to put a question mark by it. And then as I was going back through the notes, I realized I didn't understand very well what a complex virus was. It seemed to be that there was different examples. They didn't really seem similar to me. Why they were called complex. So I put some stars and question marks here on my notes about things that I didn't feel like I understood very well. There are some of these blue ink brackets on some of my notes here, and what those are from is when I went back to these notes after listening to the lecture, I found some things that were in the textbook that weren't in the lecture. So my instructor never mentioned this phrase, matrix proteins. So when I'm prioritizing my work on studying, I'm gonna, if I don't learn that particular phrase, I'm probably good. The instructor didn't cover it. So continuing with section 21.1, we have a list of types of DNA or RNA, genome, all kinds of sort of scattered notes here, right? So when we say notes, again, from the textbook, that doesn't mean copying down long sentences or paragraphs. It means writing down keywords. It means writing down lists of things and occasionally a sentence or a phrase, but you want to make sure you're always putting those into your own words rather than copying them directly from the textbook. If you can put it in your own words, it means you understand it. If you can't put it into your own words, then you are not understanding it. Now, sometimes when you're working on this and you're getting tired, it gets harder and harder to do that. So that's a good point to stop and take a break and come back to it later. You'll be able to do it when you're mentally fresh again. All right, so there's this long section about the Baltimore classifications. I just listed them and then I put a note to myself. This is confusing. I don't totally understand it. Maybe when the instructor explains this in lecture, I will be able to understand it better. I'm hoping I have a good instructor who's going to help me make sense of this. So you may not be able to understand everything from just reading the textbook. So you don't need to have that expectation for yourself, right? But you want to try and put down in your notes the sort of the level that you do understand, and then point out to yourself in some way in your notes whether you need to come back to that at a later time. Okay, so moving on to section 21.2. That I have a whole bunch of really kind of small writing over here because I didn't leave myself enough space. That I wanted to go through the different steps in replication, attachment, entry, replication and assembly, egress, or release. I didn't leave myself enough space here. I got wrote some notes in here. I might want to go back and rewrite this in a little more detail. So this little list at the beginning is actually from the textbook's learning outcomes that it has at the very beginning of the section. So these are the topics that I should be looking for in my notes. I should be able to answer a question. You know, what are the steps in replication? What is the difference between lytic and lysogenic? What happens in transmission? What are important viral diseases? What is the economic importance of viruses? I should be able to answer those questions after learning the information in this section. All right, so a little less, some of this stuff wasn't covered in the lecture. Ignore it for now. The next part of this section is the influenza example, this figure, and then in bacteria, the lytic and lysogenic cycles. I didn't write down too much here because those diagrams to me really show what the information is better. So I'd rather study this material trying to sketch out those diagrams again, rather than trying to write it out in words. So there's some more information in this section that I didn't include in the lecture, so we put that in brackets. And then a few more little facts about when, how the difference between naked versus enveloped viruses get into and out of host cells. So the ones that are enveloped need to, or they are able to fuse their cell membranes with the host cell, and ones that are naked are going to have to insert or penetrate something that goes through the cell membrane. So there's kind of some interesting differences there. All right, in the last section, 21.3, that's in my notes. What are we looking for? Prevention, treatment of viral diseases. What are some examples? And then how do we treat them? All right, so there is some information about how vaccines work in this section, although in lecture I do mention that we need to know more about the immune system and how it works in order to really understand how vaccines work. So there's a couple main ideas here. What's the difference between using a vaccine and what circumstances is it useful compared to what an antiviral drug does? Basically, how antiviral drugs work is that they inhibit viral enzymes in some way. And that the vaccine is going to be something that is going to put into your body some part of the virus, hopefully in a way that doesn't actually make you sick, but in such a way that your immune system can make a response to it. And that will mean that you will be able to fight it off much better when you are exposed to the actual live virus. All right, as viruses continue to mutate, as they do, we'll have to deal with in the future differences in vaccines that are needed, and also resistance to the vaccines, as well as changes in how they respond to the antiviral drugs. And so of all of the charm characteristics, one of the ones that viruses can do is adapt. That is, they can have changes based on natural selection. You try and kill it with a drug, you become resistant to it. Right, so I have 3 handwritten pages of notes from the textbook, having gone through, watched the lecture, done a pre-read of the chapter, didn't write any notes from that, watched the lecture, wrote notes from it, read the book, wrote notes from it. Now what I need to do is go back through all of my notes from both lecture and my readings and write myself a study guide from it. So the purpose of the lecture in general is to organize and explain the information, to focus on the things that the instructor wants you to learn and the way that they are sort of a way to think about them that fits in with our current scientific understanding. The instructor may also add context, examples, and try and put in things that are relevant to your daily life right now, and maybe even put a personal spin on, right? So if I were a research scientist who studied Ebola, I'd probably talk a lot about Ebola. Right? I don't study viruses at all, so I didn't really have any personal spin in this lecture. Right, so looking back at the overview slide from the lecture, there's 4 questions that you should be able to answer. What are viruses? What are the types of viruses? How do viruses replicate? And what are viral infections? So I wrote down my outline organized around those 4 points. I copied all of the kinds of information that I have from my notes from both lecture and from textbook into this explanation of what are viruses. Now, as you're thinking about how to answer this question, remember that there could be, say, a 1-point question about what are viruses, or some true-false questions about what are viruses. But what if you had a 10-point written question about what are viruses? You don't want to just say they're obligate intracellular parasites. That's a one point answer, right? Why is all this stuff here under what are viruses? Because if you were asked a really big points question, you don't want to just give a 1-sentence answer. You want to say sort of who, what, when, where, how, why, right? What are they? What is their structure? What are they called? What are they made of? They're a protein capsid made of capsomeres. They're going to contain DNA or RNA, some surface proteins or glycoproteins. They may or may not have an envelope. They don't have a Latin binomial name because they're non-living, right? So they're small. Why are they important? Because they cause diseases in both humans and in our economic activities related to, let's say, plant crops, right? So you want to be able to sort of go on and on about what viruses are. But if you had only a 1-sentence answer, then you should start with this. They're intracellular parasites and they're non-living. All right, when we have types of viruses, there's a couple different ways of organizing them or talking about what kind they are. So we can categorize them by shape. You should understand those differences, maybe be able to recognize a picture. They may be encapsulated or not encapsulated. And then by their genome, sort of in a simpler version, and then in the Baltimore classification. And I have a whole nother page on Baltimore classification because it was not going to fit on my 1-page outline. So this is highlighted with a star because I have a whole nother page for that. How do viruses replicate? You've got these 4 steps: attach, enter, replicate, and egress. And kind of why do viruses replicate? What are they trying to do? They're trying to increase their own numbers. They're trying to transmit to new hosts. So we have kind of a list of pieces of information. How do they do entry? It depends on the different kinds. And by the time you're writing this outline, you might understand. So if you already know the word endocytosis, for example, you just need to associate that with this concept of entry. You don't need to, re-describe this phenomenon of endocytosis if you already understand what it means. It means that a vesicle is going to bump up against the cell membrane and it's going to get absorbed, right? So if you really understand what that means, then you say, aha, encapsulated viruses are going to be able to enter their host by this thing. All right. Coming out of the host cell, they're either going to lyse or cause apoptosis happening to the cell. Again, if you don't know what that means, you need to look it up. And then budding, perhaps what are the pros and cons of doing each one? And then there's this special case where phage can do the lytic or the lysogenic cycle and some of the vocabulary associated with that. So this is an outline. This is not all the information on one piece of paper here written out in sentences. It's more like a checklist of things that I need to make sure that I understand. All right, what about viral infections? This lists here and more symptoms are caused by damage and changes in infected cells, actions the immune system takes to kill the virus. Each strain can only infect a closely related species. All types of organisms get viruses, and then antibiotics, vaccines, and antivirals. So again, if you didn't remember all of the, or understand all of the information to do with these, I highlighted it here. Maybe you need a little more information, right? But this is the one-page study guide condensed down from all that information based on the outline of the lecture. Now, as I was going through, there were some pieces of information that didn't quite fit, right? So I wrote down this sort of other list of pieces of information that I might get one question on an exam, or maybe a true-false question. How were viruses first detected? By filtration. They were called filterable particles. So that doesn't really fit into any of the other parts of the outline. It can only be imaged via electron microscopy, right? You could get a question, you know, true or false. Virus particles can be seen using a light microscope. That would be false. Enveloped viruses are more fragile. RNA and segmented genomes mutate more rapidly. So this is the kind of information that might be used for questions that were, what are the advantages and disadvantages to the virus of having some of these different kinds of structures or attributes. So these might be pieces of information you could use to support an argument if your instructor asks that kind of written questions. All right, so what are the things if you are gunning for an A+ in this class? Memorize all the virus examples, the shape, type, classification, and symptoms, right? Personally, for me as an instructor, I'm not going to ask you those kinds of questions, but you may have professors that do. You'll find that out after the first exam. All the Baltimore classifications, examples of diseases with the different types, and then all the steps of replication and protein synthesis. So this is way, if you know, your instructor would have gone through all these step by step if they wanted you to know those, hopefully. All right, so what else do I need? Well, Baltimore class, I left for another page on my notes. And I did a little bit of background information for myself over here. All cells have ribosomes, double-stranded DNA to double-stranded DNA polymerase, RNA polymerase that goes from DNA to single-stranded RNA. What viruses might need is an RNA polymerase that goes from RNA to RNA, right? Because that's different than what cells normally have, because it goes from DNA to RNA. And then they may need reverse transcriptase from RNA to DNA. All right, so what I did here is I just rewrote this information about what are they starting, what class they are, what do they start with, how do they get to a messenger RNA, right? And so they need to get that messenger RNA in order to make the proteins that they need in the cell. They may also need to do something different in order to copy their genetic material. All right, so one thing that you might start with thinking about Baltimore classification is we've got these numbers 1, 2, 3, 4, 5, 6, 7. You might have an instructor that says, what type of genetic material do class 4 viruses have? Right, so that case, you need to be able to say single-stranded RNA plus. So these are organized. There's 7. And to me, they group together as 2, 3, and 2. So the first 2 are DNA, right? The second 3 are RNA, and the last 2 use reverse transcriptase. So that's a way of organizing these in your mind. At least you know that if it's a 1 or a 2, it has DNA, one's double-stranded, one's single-stranded. And then as the categories go down, there's a little bit of an order to them, double-stranded first, then single-stranded. Except for 7, I think they were discovered last. All right, so how do we get from the genetic information to an mRNA? If you have double-stranded DNA, you use RNA polymerase and just do transcription. So this is just like a normal process in a eukaryotic cell. If you have a single-stranded DNA plus, now that means that the gene is on that strand, but you don't go straight to mRNAs. It turns out you have to go to double-stranded DNA first, and then basically get to the mRNA the same way that category 1s do. Right, so these sections, these things happening are going to be repeated as we go down this list. Double-stranded RNA is going to make a messenger RNA, a single-stranded messenger RNA using RNA polymerase, and that's a special one that viruses have, the RNA to RNA. And so on down this list. All right, so if you are pressed for time, trying to memorize this and really understand it, it's going to be your last priority, because you could probably get a C or even a B on an exam based on the lecture that I gave without memorizing this information. Okay. So how are you going to go about studying all this stuff, right? Now, having gone through all these steps, you have learned a lot of it, and that's part of the point. Some of it you should understand because you know related information already, and you're just adding a little bit more. Some of it you should understand because your instructor did such a good job of explaining it. But what are you left with, right? So go over your study guide, your 1-page study guide here. And if there's vocabulary words on here that you still don't really feel like you can write a good description of, do flashcards for your vocabulary, right? Flashcards for vocabulary is a useful thing. Write out different versions of answers to the questions: what is a virus? What types of virus are there? How do viruses replicate? And how do we recognize or classify viral diseases? Try to write out a 1-point version of answers to these questions, a 5-point and a 10-point, right? 1 point is going to be 1 sentence, 5-point is going to be 2 or 3 or 4 sentences, and 10-point is going to be 8 or 10 or 15 sentences probably, right? This is like, write everything you know about what a virus is. And run them by a study group, run those answers by your instructor. Work through all the study questions in the chapter, check your answers in the back, look up the correct answers if you are missing them, add that information to flashcards if you really feel like you don't know it very well. Practice sketching out the replication cycle in animal cells versus prokaryotic cells. Make sure you understand the difference between lytic and lysogenic. Make up at least 1 multiple choice or true/false question for each slide in the lecture, at least for each slide, right? What could your professor ask you that would be a multiple choice or true/false question? Multiple choice questions, you should write 4 or 5 answer choices, 1 of them right, and the rest of them tempting, but wrong. All right, so this is really, really going to help you. I can't emphasize it enough. I think students get confused answering multiple choice questions because they don't have sort of that reverse. If you sit down and start to try writing multiple choice questions, you start to see that there's kind of a pattern to how it's done and how the different answer choices relate to the correct answer choice. So really encourage you to practice doing that. Make up a few more possible written questions and practice writing answers out to them. If you find any of these things that you can't do or you don't understand, that's the time for your professor visit office hours, your study group, tutoring. Now, hopefully you're trying to do all this work ahead of time before the exam, so that this isn't midnight, 1 in the morning, the night before the exam, and you don't have a chance to do this. All right. Try drawing a mind map as you're working on organizing this information. I'll show you what that is in 1 sec. And then make a table of the Baltimore classifications and memorize it, or at least work through it, so that you have kind of at least some of them, you know, whether they're DNA or RNA, and then what does that mean in terms of how they're going to use that genetic material. Making a table or a summary diagram or summary table of some of the kinds of information is going to be really helpful. Don't just copy it down from your book, but make it yourself, and then fill in the answers into the table to build it. Again, doing that process is going to help you learn it. All right, so by the time you've done all this stuff, you should know most of the information, and you're just going to come back and keep going over some of those flashcards until you are sure that you have them memorized. All right, last thing is a mind map. What is a mind map? It's a diagram that comes from your mind, right? So here's my mind map of this information. And one way that it's really useful is that it's going to take kind of the topic of your lecture, and you're going to put that in the middle of the paper. And then somehow around the main topic, you're going to put the different subtopics. So it's like a visual outline. What are the things that we talked about? Types, how they do replication, and how do they form infections. And part of the purpose of the mind map and why it can be a really useful tool for studying, especially to be able to write answers to questions that ask you about how do these things relate to one another, right? Or compare and contrast, or give the pros and cons, right? And that's what the mind map is intended to show, not just lists of information, but how it relates, right? So we've got the types of viruses, and we're going to write down some of the information. You don't want to put all the information on here, because what's more important is the relationships between the information. And so this isn't the same thing as a comprehensive study guide. It's more a way of you thinking about the information or doing active learning. The process of drawing the diagram, there's no one right diagram. It's just a process to help you think about how the information relates. All right, so I've got some of these different Baltimore classifications written down. I've written down some of the main parts of replication: attachment, enter, egress, 2 different kinds of replication, phage versus animal. What does it have to do during replication? It's going to make proteins, it's going to make copies of the DNA or RNA. How do we know? What is it? How does it do that? Well, it depends on their Baltimore classification. So that's how these pieces of information relate to one another. Phage has the lytic and lysogenic cycle that's similar to something that these reverse transcriptase viruses can do, which is hide in the host genome. Right, so that's actually a comparison that might not have been brought out in when you're actually looking at this information in any other way. Right, so what do things depend on? Right, how the attachment happens depends on the type. How the egress happens depends on the type. So when we have those types listed over here, you can see how it relates to how they're going to do something in particular. Right, and then what happens with infections? We're going to have one branch here. What are the symptoms? How is it transmitted? These things go together. And then what are the clinical or medical implications? And we're going to worry about prevention. We're going to worry about treatment. So this is another way of organizing the information. I got a little goofy by the end. I was drawing a needle going into a person's arm with the frowny face. I was looking at this drawing just now before I started this recording, and at first I couldn't figure out what it was that I'd drawn. Right, we can't treat viruses with antibiotics, and which things relate to one another. All right, so this is just my mind map for this information. Yours doesn't have to look exactly the same. I hope that what we've talked about has helped you understand when your instructor says you need to study, like, what do you do? What are those processes? So it sounds like a lot of going back over the same information, writing it down. I promise you that that is how we learn, and that once you have gotten more into a habit of planning ahead with your times, if you have the time, so that you can do all these steps, this is what it's going to take to, get a really high A on a regular sort of traditional type of biology exam at a higher level. This is not for a GE course. This is for an upper level course, 200 or higher. All right, I hope this helps you do good in your classes, and you certainly learned a bunch about viruses.